Hello everyone and welcome to today's webinar, Serum Autoantibodies Differentiate Rheumatoid Arthritis Subgroups. I'm Dr. Adrian Churchman, Director of Europe and North America at Sengenix, and I'll be your moderator for today's events. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them in the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Also, if you have experience of technical questions, you can utilize this Ask a Question box and we will assist you directly. Today's educational web seminar is presented by LabRoots and is brought to you by Sengenix. Our mission at Sengenix is to empower you with biologically relevant and actionable immunoproteomic insights across a broad spectrum of diseases. We believe that these insights are possible through highly specific and reproducible antibody biomarker signatures that are measured using correctly folded proteins. We achieve exceptional precision for discovery and validation of autoantibody biomarkers using multifaceted strategy, from the way we design our protein microarrays to how we analyze the data. Our proprietary CREX technology ensures that the proteins displayed on the microarrays are full length and correctly folded, preserving the 3D conformational epitopes that 90% of our antibodies bind. Sengenix offers custom and ready-to-use protein microarrays for high-throughput autoantibody profiling, including sample and bioinformatics analysis. You can also access our technology and services through distributors and certified service providers. To learn more, visit sengenix.com. Without further ado, I'd like to welcome our speaker, Dr. Jaeyun. Dr. Sung is a computational biologist at the prestigious Mayo Clinic, where he serves as a senior associate consultant and assistant professor in the divisions of surgery, research, and rheumatology, and the Center for Individualized Medicine's Microbiomics program. Dr. Sung's research focus on developing computational tools to advance precision medicine for autoimmune inflammatory disorders. Jay, I hand over to you.
Thank you for the kind introduction, Adrian. Let's see here. Okay. All right. So I believe everyone is looking at my title slide right now. And the title of today's talk is going to be Serum Autoantibodies Differentiate Rheumatoid Arthritis Subgroups. As Adrian mentioned, I'm an assistant professor in the divisions of computational biology and rheumatology here at Mayo Clinic, which is located in Rochester, Minnesota, in the US. Okay. And today I'm going to be talking about some recently published work in our group using the Sengenix platform. And this paper was published in Scientific Reports last year. And the title of the study is shown on the slide. And this was led by a PhD student in our group, Kevin Cunningham. So special thanks to Kevin. Okay, so the title of the paper states, patients with ACPA positive and ACPA negative rheumatoid arthritis shows different serological autoantibody repertoires and autoantibody association with disease activities. Now in the next few slides, I'm going to dissect what all these terms mean in case there's people that the audience members who are not familiar with seronegative, seropositive, and also the autoimmune inflammatory disorder rheumatoid arthritis. Okay, so first, rheumatoid arthritis. It's a systemic autoimmune inflammatory disorder characterized by joint inflammation, mainly characterized by joint inflammations, but it also causes inflammation in the heart, the spine, the eyes as well. And its symptoms include chronic swelling and pain, and also bone erosion and joint deformity, damage to other organ systems, such as I mentioned, the heart, eyes, and also the lungs as well. Here in the US, we have an estimated approximately 1.5 million adults with an RA diagnosis. And one of the clinical hallmarks used as a biomarker for rheumatoid arthritis is the blood IgG anti-citrullinated protein antibody, or ACPA. So it's and I'll talk about this in the next few slides, but it's an antibody developed by our own immune system that goes after citrullinated proteins in our body. And it's one of the most commonly tested serological biomarkers for rheumatoid arthritis. So For those who don't know what an autoantibody is, these are antibodies that target macromolecules in cells of our individual's own body. It can cause direct tissue damage and dysregulation of normal cellular processes. And it could also be used as biomarkers for several autoimmune diseases. For example, there's ACPA, as I just mentioned, in rheumatoid arthritis. There's rheumatoid factor as well. And there's also ANCA or anti-nuclear protein antibodies in vasculitis. So depending on the autoimmune disease, there are hallmark biomarkers for that disorder. Okay. So speaking of ACPA, which is again the biomarker for rheumatoid arthritis, it's a major risk factor for developing RA. It's involved in various disease mechanisms, which I'm summarizing in the figure on the right. I don't have time to go into all these intricate details, but it is involved in various disorders such as T-cell, various disorders in rheumatoid arthritis, such as blocking the T-cell, B-cell communication, causing harm to bone tissues and also the synovium in the joints. Now, this biomarker, it is used as a common serological blood test for rheumatoid arthritis. It has high specificity against RA, but really low sensitivity. And I'll talk about in the next slide what that really means. Okay, so in this table here and this is on about 300 or so, close to 300 RA patients with 100 healthy subjects. Now, these numbers are a little bit outdated, but the conclusion still remains robust. And here's what I mean. So what you're seeing in this table are the different, for the columns, are the different biomarkers, antibody biomarkers that I used to clinically diagnose rheumatoid arthritis. And on the rows, where you see cutoff specificity, these are just performance metrics of how well you can actually classify rheumatoid arthritis. So if you look at the top row where I'm highlighting in the blue, specificity, that's basically, okay, if you're healthy, are you negative for ACPA? 99% for the IgG type, which we use here at Mayo, 99% that's true. Okay, for the sensitivity, which means if you have rheumatoid arthritis, if you truly have rheumatoid arthritis, do you have ACPA? And the answer is only 60%. So this is what I mean by ACPA has great specificity against RA, but I mean, not so great sensitivity of ACPA as a diagnostic marker. So here, now I won't go into basically why that is the case, but for the purposes of this talk, there's two concepts that the audience needs to know, which is just seropositive and seronegative. So here, that just means ACPA positive RA patients who have ACPA, and ACPA patients who are seronegative, and RA patients who do not have ACPA. So ACPA positive and ACPA negative rheumatoid arthritis, they're all clinically diagnosed the same. And what is commonly known or thought of in the field, and this does not turn out to be completely true, but what's commonly accepted knowledge is that the only difference between these patients is that they just have one subgroup has ACPA in their blood, the autoantibody for citrullinated A proteins, and one subgroup doesn't. Okay. Now, just for terminology purposes, seronegative, that just means you just don't have that biomarker in your blood. So you test negative for that specific autoantibody. So clinically, the person does not have the same antibodies compared to a person who is seropositive, although they are still diagnosed as the same disease. And seronegative conditions include most autoimmune disorders, such as rheumatoid arthritis, ankylosing spondylitis, and so forth. Okay. Now, a clinically unmet need here is that there is no standard laboratory test for diagnosing seronegative RA. So these are patients who, if they have RA symptoms and they're positive for ACPA, then it's not very hard for the clinicians to diagnose them as as RA, excuse me. But if they don't have ACPA, what ends up happening is they go to this clinic, to that clinic, whether it's spondyloarthritis, whether it's osteoarthritis, whether it's vasculitis, and they bounce off from specialty to specialty, and they come back as being negative for all of those other diseases. They still have RA, and they end up being diagnosed eventually as RA. 2 to 3, on average, 2 to 3 years later after they first showing symptoms. So, and that's a problem because it hinders timely interventions, it hinders when they start on treatments and so forth, and also causes, because during that lag of 2 to 3 years on average, and they're not getting the treatment that they need, the patients have an increased risk for severe disease forms. Importantly, there's a rising, this is a result that we found by my colleagues here in the Division of Rheumatology, but there's been a rising proportion of seronegative, specifically ACPA-negative RA cases. So, as I mentioned before, what's commonly understood is that rheumatoid arthritis patients who do and who don't have, who are ACPA-negative, ACPA-positive, they're assumed that the only difference is that one has ACPA in their blood and one does not. So the difference is explained only by a single molecule. That does not, more and more evidence is coming out in the literature saying that is not true, actually. So recent reports of differences have been found in actually the disease course and treatment response patterns. So patients with ACPA-negative RA, so seronegative RA and seropositive RA, they're actually responding a little differently to standard treatments. They have differences in the risk allele frequencies in the HLA region. Differences have been found in gut bacterial species, differences in their proteins in their blood, and also differences, well, highlighted by our study that I mentioned in the opening slide, their serum IgG autoantibody repertoires are different. So in the field of rheumatology, some clinicians are thinking that, yes, it is the same symptoms of RA, but they could be underlying two distinct diseases here, serum ACPA-negative and ACPA-positive RA. So this is a big field of interest of mine and of our laboratory here at Mayo, and something we've been actually heavily studying for the past 2 to 3 years. Okay, so really to elucidate the differences between ACPA-positive and ACPA-negative RA, besides just the obvious ACPA, we need more studies in the seronegative subgroup of RA. So, and first of all, ACPA-negative RA is a relatively understudied subgroup of RA. Most RA studies out there only look at the ACPA-positive subgroup, but the ACPA-negative group, you know, there more and more evidence is coming out that the proportion of ACPA-negative RA is rising. They respond to treatments differently, difference in gut microbes, proteins, etc. So basically, we need more research into this ACPA-negative RA, this neglected group of RA patients. And also the molecular, cellular, and immunological differences between these two subgroups are not well understood. So this is really the impetus of our study. Okay. All right. So you may be asking why autoantibody profiling. Why do we specifically use the Syngenix platform for our study here to look at the difference between ACPA-negative and ACPA-positive? Well, we wanted to study, before we even got to know the Syngenix and the products they offer. In my group, we wanted to study humoral immunity, not just, we don't study also the antibodies and autoantibodies, we also study a little bit of the complement system as well. But anyway, we want to study humoral immunity at high throughput scale. Being a computational lab, we look at high throughput data sets, all these different omics data sets, and we wanted to see what omics technologies could bring in the autoantibody profiling space. Also, autoantibody profiling is good to obtain novel insights into potentially so if you find good hits, you could find potentially pathogenic or disease modulating processes. And we're very intrigued by the fact that every patient in RA, and not just patients, but even healthy people as well, they have different antibody or autoantibody signatures, which basically summarizes their entire immunological history throughout their life. We find differences in, and this is a study that won't be published, that I won't be talking about, but we have published, is we find that that autoimmune inflammatory disorders, patients with autoimmune inflammatory disorders, specifically RA, have differences in their autoantibody repertoires compared to healthy controls. It's also looking at autoantibodies. It's a promising search space for clinical biomarkers. Most, but not all, most autoimmune disorders do have a bona fide biomarker and it does take the identity of an antibody or an autoantibody. If you know if you identify the autoantibody, then with further characterization, or sometimes by default, you know the autoantigen. So basically, what the body's antibodies are attaching to or targeting, and that could lead to autoantigen targeting therapies. Okay, so I hope I explained the motivation of why we want to do autoantibody profiling in the first place. Okay, so the key questions now, coming to the crux of the results I'm about to show you today, is so the key questions we aim to address are, I'm just enumerating them here, but are there differences in serum autoantibody abundances between ACPA positive RA and ACPA negative RA? Second, which serum autoantibodies in the two RA subgroups are either higher or lower in abundance compared to healthy controls? Third, which biological functions are the autoantibody targets involved in? Okay, so what processes in the human body are these autoantibodies that we found to be differentially abundant? Are they targeting? Are they messing up? And do any of the circulating autoantibodies correlate with disease activity in RA patients? Okay, so the main objective is twofold. It's to show that patients with ACPA positive RA and ACPA negative RA display differences in serological autoantibodies and also autoantibody associations with clinical disease activity. And I hope in the next few slides, based on our results, I'm able to convince members of the audience that we pretty much succeeded in the aims that we set out to accomplish. Okay, so first, here's our study design. I apologize, but I can't use the mouse here, so I'll have to guide the audience eyes through by telling them what direction on the screen to look at. Okay, so if you look on the top left, this is just a summary of the patient cohorts, okay, study participants. So we have 32 patients with ACPA positive RA, 30 patients with ACPA negative RA, and 30 controls, okay? And among those 92 people, we take their blood, specifically serum, and with a serum sample, we use the Syngenix iOme discovery protein microarray, which I'll talk about in the next few slides. And this allows the detection of 1,622 IgG type autoantibodies, okay, for all of the samples, for all of the patients. Okay. All right. So why use the Syngenix iOme discovery protein microarray? Well, it's quantitative profiling of over 1,600 autoantibodies, so much more high throughput than what you could do with ELISA or Western blotting. And what's really interesting about their technology is that the autoantibodies target full-length, correctly folded native human proteins. Okay, so it's not targeting misfolded proteins, damaged proteins. It has to be full-length, correctly folded native human proteins. And this really ensures high specificity between the autoantibody and autoantigen interaction. All right, so their platform, Syngenix platform, this is just a high-level summary of how the microarray technology works. Now, it's essentially a microarray. In step one, you have protein antigens immobilized on a slide of a chip, and then step two, in step one, you apply patient serum. In step two, some autoantibodies in the patient in the subject serum or plasma will stick to the autoantigens, and then there's a washing step. And step three, now with the autoantibodies from the patient that's stuck to the chip, the ones in the yellow, you have another autoantibody that goes after those specific autoantibodies, and that antibody in blue is attached to a fluorophore. So basically, the yellow ones are what we want to target, but the blue antibodies attached to a fluorophore is what's going to output the measurement in the microarray that we can quantitatively analyze. Okay, so this is just a really high-level summary of how the technology works. This is all on the company website. So for those really interested in the weeds of how this technology works, I recommend going and reading their summary. Okay, so how did the data look like? Okay. So first, so to summarize again, to remind the audience again, we have autoantibody profiles from 92 study participants, and each autoantibody profile has 1,622 IgG autoantibodies that it could measure. And we put all those samples, those autoantibody profiles, into a principal component analysis plot. So I'm pretty sure people have seen PCA plots before, basically reducing all that dimensionality of 1622 into just 2 dimensions. And you can see basically the heterogeneity across the 92 profiles. So in the PCA plot on the right, the red are the ACPA positive RA autoantibody profiles. The gray are the ACPA negative RA antibody profiles, and the blue are the samples or the autoantibody profiles from the controls, healthy controls. Okay, and you see a lot of heterogeneity in the controls, a lot of and some heterogeneity in the ACPA negative RA. Interestingly, there's a lot tighter. There's very small heterogeneity in the ACPA positive RA. It could be because the ACPA positive RAs patients are are more homogeneous compared to ACPA negative RA or controls. Okay, but this is what we see in a very high level overview of the data. Okay, and this is what's called a ternary plot. And what we do here is we take the mean abundance of a particular autoantibody, and you can get the mean for that in the control, mean of that in the ACPA negative RA and the ACPA positive RA cohort. You kind of scale it. So scale those means, those 3 different means, so it sums to 100. So if everything is the same in the 3 different study cohorts, that means 33%, 33%, 33%. It's dead in the middle. Okay, so the dots here, the dots in the previous slide here, the points each represent an autoantibody sample. It actually represents a person. But here, the points represent an autoantibody. An autoantibody, it's scaled abundance of an autoantibody in comparison to all study groups. So the focus should be not on the dots in the middle, but it should be on the dots towards the corners of the ternary plot here. So the closer a point is towards a specific corner, so let's say the top corner, ACPA positive RA. So there's a point, I guess, closer than everybody else towards ACPA positive RA. Well, that means that autoantibody is higher in ACPA positive RA compared to everybody else. Okay, and there's a point that's going in, let's say, the 4 o'clock direction. That means it's higher in controls than the RA subgroups. And you can say the same thing for ACPA negative RA. Okay, so basically what we're seeing here is we do see differences at a very high level. We don't know what the specific autoantibodies are yet, but we do see differences in the profiles between ACPA positive RA, ACPA negative RA, and control. So this is kind of our entry point into, hey, there may be something interesting here. There may be specific differences. Let's go a little bit deeper. And we do that in the next slide. So what we're showing here, and I apologize for the small text, but what we're showing here is compares differentially abundant autoantibodies between ACPA positive RA, the blue, and the red, and the controls, blue. In the next slide, I'm showing differentially abundant autoantibodies between the ACPA negative RA and the controls. Okay? And specifically in these 2 plots, I'm looking at those autoantibodies that are higher in RA subgroup compared to controls. So to summarize, there were 22 autoantibodies higher in ACPA positive RA compared to controls, and 19 autoantibodies higher in ACPA negative RA compared to controls. Okay. And there was 1 overlap, anti-GTF2A2. And by the way, anti is just a symbol for antibody. So what that means is there's an antibody that's going after the GTF2A2 protein. Okay. Now we want to look at autoantibodies that are actually lower in an RA subgroup compared to controls. So in other words, higher in healthy people compared to RA. Okay. And we find that 30 autoantibodies are lower in ACPA-positive RA compared to controls, and 25 autoantibodies are lower in ACPA-negative RA compared to controls. And there was a lot more in common in the lower batch. So these proteins that you're seeing in the screen in the gold text, these are the proteins that were found. These were the autoantibodies that were commonly found to be lower in the RA subgroups than controls. So we find these autoantibodies, okay? Now, what processes are they targeting? So what we did next is we looked at the autoantigen targets that these autoantibodies are targeting, and these specific proteins, and we did a functional enrichment test to see, okay, what are these targets involved in? Okay. And we find that the autoantibody targets are functionally enriched in various biological processes. Here in A on the left, these are enriched gene ontology biological processes of the targets of the autoantibodies that are higher in RA subgroups than controls. So the blue represents ACPA-positive RA, the orange represents ACPA-negative RA. So to summarize, these are the enriched biological processes of the targets of the autoantibodies that we found to be differentially abundant. So in the ACPA-positive RA group, you see a bunch of cell death, apoptosis, programmed cell death. Those are the top hits. So basically in ACPA-positive RA, those people have have autoantibodies that are targeting cell death related matters. In the orange, we see DNA template transcription, cellular responses, so RNA synthesis. So regarding gene expression, regarding RNA metabolism, possibly, is the processes that the autoantibodies are targeting ACPA-negative RA. Now you can tell the same story in the autoantibodies that are lower in RA subgroup, but I'll just abbreviate that for now. All the results are in the paper. Okay. Now, what we did next, our final question was, does the autoantibodies correlate with clinical disease activity? Okay. So what we're looking at here is we're looking to see, based on the ACPA-positive RA group, on the left column, the ACPA-negative RA group in the middle column, and just putting those subgroups together, all RA in the right column. And what we're doing here is for those people, for those patients, we're taking their autoantibody abundances that we found in their serum, and we're correlating that with their disease activity. So in rheumatoid arthritis, there's multiple indices, quantitative indices that we use to approximate the patient's disease activity. Some people use something called DAS28. Some people use, some providers use DAS28-CRP, and some people use the SDI. Some people use the CDI here, which stands for Clinical Disease Activity Index, and that's one of the often used indices for disease activity for rheumatoid arthritis, at least here at Mayo. So basically, these are just correlation plots. In these bubble plots here, the columns represent the study group, the rows represent the specific antibody that we found to be significant, autoantibody found to be significant. The size of the bubbles represent the significance of the correlation. The colors of the bubble actually represents the Spearman correlation, so the magnitude of how well things are correlated. Okay, red, the more red means positively correlated. More blue, darker blue, means there's a negative correlation between autoantibody abundance and disease activity. Okay, so to dissect this, what really stood out here, and also the stars represent those that, the bubbles with the stars represent those that we found to be statistically significant. What immediately stood out to us was how different ACPA positive and ACPA negative look. Okay, so in the ACPA positive RA column, you find 11 autoantibodies that are significantly correlated with clinical disease activity index. Okay, and in ACPA-negative RA, you find 15, and there's no overlap. So what this shows is that when it comes to measuring disease activity and specific autoantibodies, there's actually quite a stark difference between these two RA subgroups. And we find only one on the bottom right for anti-CISH that is correlated in in both subgroups with disease activity. Okay, so the conclusion here is that serum autoantibodies display subgroup-specific correlations with RA disease activity. And this is just taking one example, CISH, anti-CISH, which was the autoantibody found to be correlated with disease activity in both subgroups. When you combine the subgroups into just one homogeneous cohort. And this is the correlation between CDI and the abundance of anti-CISH. Okay. So take-home points here. We profiled serum autoantibody abundances, serum autoantibodies in two subgroups of RA, so the ACPA positive and ACPA negative RA subgroups. We observed serological differences between the two RA subgroups. We found autoantibodies were found to be that were higher and also lower in disease. Surprisingly, there was little overlap between the RA subgroups. Autoantigens, so the targets of those autoantibodies were functionally enriched in programmed cell death, RNA, and also nucleic acid, also other nucleic acid DNA metabolism, cell signaling. And we find that autoantibodies correlated with CDI, but that correlation associates differently depending on the ACPA status. And what we mean by that is, yes, we do see correlations, but the correlations of that specific with disease activity, but the specific autoantibodies turns out to be very different compared to if you compare ACPA positive and ACPA negative RA. Okay. And last, basically our data motivates future studies of ours and hopefully others for stratifying RA patients based on their molecular and immunological profiles. So someday in the future, when a patient walks into our clinic, we'll be able to take their autoantibody signature or their autoantibody fingerprint. And based on the profile we see, we can tailor their treatments based on their molecular patterns. That's the eventual goal. So before I end, I'd like to acknowledge a few folks. So first, my group here at Mayo Clinic, we're the Computational Systems Medicine Research Group. Kevin Cunningham, who's a PhD student in our group, who led the study that I presented to you today. Also, my clinical collaborator and mentor, Dr. John Davis, who's now the chair of the Division of Rheumatology here at Mayo, and funding sources for this specific project. And that is all. And I believe there's a poll question that we need to ask. So, Adrian?
Yes, thanks a lot, Jay. That's a really informative presentation. And as you say, before we jump live into the Q&A portion of the webinar, we'd like to ask the audience a question. Please take a moment to rate how important you think autoantibody profiling is in research. And you should be able to click the slide and just put in your answer. We've still got some responses coming in, so I'll give it a couple more seconds. Right, I think I'll show that to everyone in the audience, but we've got, I think, a very important rating of 81.82%, which is excellent. Thank you very much, Jay. Now we're going to start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located to the far left of your screen, and we'll ask as many questions as we have time for. So let me just have a quick look at the box. Yeah, we've got a few coming in already. First question is, were patients treatment naive?
Yeah. No, they were not. Now, in the ideal world, we would have all these patients to come in as treatment naive, with basically not having any eliminating the source of confounders. In our case, this was a retrospective study, so we just used the serum that was available in our RA biobank. What I do need to mention, though, that they were not on any antibody inhibitors. So they're not on, for example, anti-CD20 inhibitors such as rituximab or ocrelizumab. So they were on treatments like steroidal treatments. They were on in non-steroid treatments, but not on treatments that are known to specifically target antibodies. Because that, if so, that would basically eliminate the rigor of our study and be a huge confounding factor. I hope that helps.
Thanks very much. We have another one come in. Can an ACPA positive patient become ACPA negative?
I have, based on my knowledge, I have never seen that. Once you have that, because autoantibodies in general are very stable. So the vice versa happens. The opposite happens all the time. ACPA negative patients, so patients with RA who are negative for ACPA negative, eventually over time they can, but not always, they can develop the antibody. But being positive for ACPA, if you have RA and then suddenly it disappearing, I've never heard of that case.
Okay, just while we wait for a couple more to come in. From the presentation you gave, obviously autoantibodies are tied to diseases. I saw that you had some data from many autoantibodies are elevated in your controls. Could you explain that a little bit for everyone, please?
Yeah, so that is a very interesting question, because traditionally, so to restate that question, that just means, why do you see autoantibodies that are higher in controls? I thought autoantibodies are bad. Well, that's not as simple as it seems, because traditionally, I do acknowledge that autoantibodies, so this is going back to my previous slide, autoantibodies are associated with disease, autoimmune or inflammatory disorders. That's why they're used as biomarkers, right? But, and I always carry, for this talk, I always carry this slide here, just to show that of previous literature evidence that, hey, even healthy people, hopefully the majority of, if not all of everyone in this audience, do carry natural autoantibodies. And what are natural autoantibodies? They're produced by the immune system to intentionally target self-antigens without triggering undesired autoimmune disorders. They actually have protective benefits. For example, they regulate normal homeostatic processes, clear cellular debris, even catalyze enzymatic reactions. By the way, autoantibodies can catalyze metabolic reactions. And also, they protect against self-antigens that may induce immune response, highly individualized, and mainly they could have one interesting future direction of study would be seeing if any of these natural autoantibodies, again, these are autoantibodies made by the adaptive immune system, B cells, specifically the plasma B cells, they make even though we feel fine. So possibly these natural autoantibodies may have a possible therapeutic role. So we believe that the autoantibodies that we found to be actually, we haven't proven it yet, but we find that the autoantibodies that are higher in controls, or vice versa, lower in the RA subgroup that you see on the right here, on the right hand side, they could be, we are tapping into the natural autoantibody space. So really, this study opened up a lot of questions in our group of wow, I mean, there is natural autoimmunity. What are these natural autoantibodies doing? Are they what kind of protective role do they have? Are there sex-related differences? Do we develop or do we diminish these natural autoantibodies as we get older? So this is a series of questions that we had that we would not have had if we did not use the Syngenix platform. Okay, thank you.
Thanks a lot for that. We have one question. IgG was reported. Were IgA autoantibodies also evaluated?
Okay, so thanks for the question. In the Syngenix platform that you can see we briefly describe on the study design figure, that only identifies IgG autoantibodies.
Right, one more. After having the diagnostic, would you have treatment based on diet? Since the ACPA positive is about attacking citrulline, can we decrease the arginine intake to decrease the state of disease or even heal?
So after having the diagnostic, you mean so the diagnosis of RA, would you have treatment based on diet?
Yeah, I
Okay,
think
so,
this is the diagnosis based on ACPA positive.
sure. Since patients are ACPA positive, is about attacking. Since the autoantibody is about attacking citrulline, can we decrease arginine? Well, that's a very tempting, in my view, a tempting statement to make. And I believe some investigators are actually looking into that question of, does arginine intake really change things? I don't know on that. We don't do food related studies. I don't think there's any studies out there proven to show that dietary intake specifically for arginine that would actually be helpful for rheumatoid arthritis symptoms. But that doesn't mean healthy lifestyle, healthy diet helps. Of course it helps. Yeah, stay away from processed foods, processed meats and all, and sugars and all. But when it comes to the particular question of arginine supplementation, I don't know how you would do removing arginine, just because it's a essential amino acid. But that is dietary measures for that particular question. I've not seen in the literature, to be proven at least, that it works.
We've got one person's asked 3 questions, so 1 question and then 2 follow-ons, and I'll just go through them in order. The first one is that can all the autoantibodies of interest be detected using the platform?
Can all are all the autoantibodies of interest can be detected using the platform? Well, remember, this is an untargeted approach, so we don't come in thinking we're going to target these specific autoantibodies. We just use what's available in the autoantibody microarray chip that Syngenix provides, and this is usually the case for these untargeted other omes. For example, if you're familiar with proteomics from Olink or SomaLogic, we look at their data. If you look at metabolites for Metabolon, for metabolomics. We don't come in with a specific hypothesis of we're going to only look for these proteins. Now, I believe Syngenix, they actually make, now they actually come up with a product that looked for the citrulline. So autoantibodies that target citrulated proteins. That's something we have a lot of interest in. Okay, but specific autoantibodies.
That's exactly right. We can do that. We can look at citrullinated proteins.
Yeah.
We can also create custom arrays. So if there is a protein of interest, we can express that and put that directly onto a custom array itself, or add it additionally to the IM discovery array. So no protein is normally out of reach. We have a very good level of expression and a good set of reliability for getting the proteins onto the array. So if there is something of interest that's not already on our list, just let us know and we'll look into it. Another question, follow-up question here is, have you teased out a few key autoantibodies and tested them quantitatively? So this is a validation question.
Yeah, so not yet. Now, obviously, that is the next step, whether to look for these autoantibodies in patients' sera using Western or just a common ELISA. We haven't done that yet. We do have plans um, for that, though. It is important to, once we find something. I mean, I do recognize the high importance of once you find something using data-driven approaches, such as these high throughput measurements, you have to validate somehow. Fortunately, validating proteins in these autoantibodies, it's not that difficult to do. So.
And then the last follow-on question was, any potential for prognosis using some of the autoantibodies, maybe a panel?
There is potential. Now, we haven't tested for that yet because we just don't have. When it comes to prognosis, rather than prognosis for an autoimmune disorder, what would be better is maybe prognosis regarding what? Prognosis regarding drug response, prognosis regarding disease activity. Autoimmune disease is one of those that just fluctuates. They're characterized by flares. So basically, if you talk to patients, they would say their flares come and go. So it's really hard to set a prognosis for that. It's different it's different than cancer, let's say. So there is potential. We haven't done it yet, and the right study design has to be in place for that.
Was HLA type associated with the profiles?
We don't have that genetic information of these patients. Now, that's something we could do, going back to the genome sequences of all those patients, but we did not look at HLA type.
And then there is another one, arginine. Nitric oxide improve vasculitis, question mark?
Improve vasculitis. Well, vasculitis is a different disease, but it's still a rheumatic disorder. I actually do not know the answer to that one, whether arginine and nitric oxide has potential in alleviating symptoms of vasculitis. By the way, it is a different disease, but it's also a disease that we do in our group. So vasculitis, for those who don't know, it's just an autoimmune disease that targets the blood vessels, sometimes in the temporal, in the main aorta, or sometimes the arteries in the head and the eyes. So a common disorder is blindness, actually. Very painful disease. Okay.
And going back to the diet question we had earlier, a diet rich in carbohydrates and low in protein could improve the symptoms, as I saw in some patients. I think that's more of a comment than a question, but would you like to go back on that?
Yeah. Which carbohydrates? Are we talking about simple carbohydrates, complex carbohydrates?
Just as a diet rich in carbohydrates.
Yeah, rich in carbohydrates and low protein. I don't understand the low protein part. It probably does improve symptoms, but getting into diet, it's very difficult because people, when I see dietary studies, I'm pretty sure the investigators all work very hard on it. But when it comes to interpreting results, in my view, it's a well-known fact that people respond to the same foods differently. I'm not sure, I don't mean this to be any disrespect to you as a provider or the patients, but is this not anecdotal? Is this not happening? I mean, can you actually generalize these claims? I'm not really sure. But what I do know for sure is cleaning up the diet, like I said before, removing processed foods, going back to the natural way, the paleo way. There's growing evidence for that, but that's not the topic of today's talk. Maybe for a private discussion over a beer.
Do you have a plan to use your 1622 autoantibody panel to test the serum of patients with other diseases and look for correlations?
Not necessarily at the moment, but it definitely can be done. The thing is, you have to be really, so just to add a little bit of context to our group, we're a computational biology and translational bioinformatics lab that look at multi-omics data to find biomarkers and to characterize disease processes in autoimmune and neurodegenerative disorders. We don't look only at these autoantibodies. We look at transcriptomics, we look at single cell RNA, we look at proteomics, we look at metabolomics. Anything that really, we try to look at as much data as possible, because disease processes, they're multifaceted. They multifaceted. So they, you know, obviously, as people know, it's not disease. Complex disorders is not just. Is someone typing? All right, thank you. I mean, it's not just a like a metabolic issue. It's not just an autoantibody issue alone. If the study design and the biological interpretation could make sense, that's when we engage in these omics technologies. For example, that's a long-winded answer, but there's nothing stopping anyone from exploring whether autoantibodies may be a key factor in diseases that are not autoimmune in nature. For example, metabolic diseases, cancer.
I think we're coming to the end here. We've not got many other questions coming in at the moment. I'll give you a little bit more time, but then I think we'll wrap up for this afternoon, this morning, Eastern time. Wait to see if there's anything else coming in. No. Well, thank you again, Jay, for your time today and the important research and giving over your presentation. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. If you didn't write a question today, but you do have one, please feel free to reach out to the speaker or to the organizers, and we'll see if we can get that done. The webcast can be viewed on demand. Lab Groups will alert you via the email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thanks. Bye.